Stem cells are unspecialized cells that are able to differentiate into many types of mature cells with special functions, such as heart muscle or insulin-producing cells in the pancreas. This aids the body in healing and organ repair. There are primarily two types of stem cells, embryonic and somatic. Embryonic stem cells can be found in fertilized human embryos and are considered pluripotent, meaning that they can be specialized into any kind of cell. Somatic cells are found in adult bodies, such as in our bone marrow, and are considered multipotent, meaning that they can only specialize into related cell types. Therefore, embryonic stem cells are much more versatile and efficient. While stem cell research has been conducted since the 1950s, using stem cells for therapy and medical procedures are a recent development. In 1968, the first successful bone marrow transplant took place, while in 1998, the first human embryonic stem cell line is produced. Stem cell research has the potential to cure diseases and save lives. However, they remain a controversial subject due to the process of destroying an embryo.
Now that we've discussed what stem cells are, we can look at the types of stem cells and where those types are found. There are three main types of stem cells: totipotent, pluripotent, and multipotent. Totipotent cells can develop into any cell that forms the body or the placenta. Pluripotent cells can develop into any cell that forms the body, and multipotent cells can develop into more than one type of cell but are more restricted than pluripotent cells. The reason why only two of these types are shown is because the only cells considered to be totipotent are embryonic stem cells within the first couple of cell divisions after fertilization. Now, embryonic cells are normally harvested when the embryo is 3 to 5 days old, and at this point the embryo is called a blastocyst. Perinatal stem cells can be found in three main locations, the first being amniotic fluid, the second being umbilical cord blood and tissue, and the third place is placenta blood and tissue. Adult stem cells can be found in a wide variety of tissue, but only in small amounts. So adult stem cells can be found in fat tissue, liver tissue, skin, bone marrow, muscles, etc. Again, only in small amounts. Now, induced pluripotent stem cells fall under the category of adult cells, and this is, again, these are have only been looked at recently, around 2007, and these involve taking adult cells that have already differentiated and reversing that process. And how this works is these cells are forced to express genes that help them maintain embryonic stem cell-like function. So the differentiation process here is being reversed. No, we're not talking about the mathematical operation. We are talking about cells becoming specialized in the body. These are basically taking adult cells and making them behave like embryonic cells because they're pluripotent. So this presents a question: how do cells differentiate? And that's actually the next topic, differentiation.
Differentiation is the process of a stem cell maturing into a specific type of cell. In the process of differentiation, stem cells activate certain genes in certain areas of your DNA in order to develop into different types of cells. Now, transcription factors, which are usually proteins that are involved in the process of converting or transcribing DNA into RNA, play a large role in determining which genes are expressed, which ultimately determines what type of cell is formed. For example, skin cells have had different genes expressed compared to stomach cells or muscle cells.
There are two types of transplants: There are autologous stem cell transplants and allogeneic stem cell transplants. Autologous stem cell transplants. Doctors use the patient's own stem cells to treat the cancer. Firstly, doctors collect the patient's stem cells from their blood and freeze it. Next, the patient undergoes chemotherapy. Subsequently, the medical team thaws the stem cells and injects the stem cells back into the patients through IV. It takes about 24 hours for the stem cells to reach the bone marrow, where it begins to grow and multiply. Thus, the bone marrow is aided in the production of healthy blood cells, and the patient is treated. Allogeneic transplantation. Firstly, doctors use a donor's stem cells instead of the stem cells of the patients. However, the donor's bone marrow must match the patient's bone marrow because the leukocyte antigens within the donor's bone marrow must match the patient's leukocyte antigens. Antigens are substances that trigger an immune response from the body. The antigens within the body are distinguishable from foreign antigens and therefore do not trigger a response. The donor's antigens must be the same or close to the same as the patient's antigens to prevent immunotrigger. This prevents graft versus host disease, where the healthy cells from the transplant attacks the patient's cells. Once the patient receives a donor, they receive chemotherapy. Next, the patient is injected with the donor stem cells through IV. This is done as soon as possible after chemotherapy because the donor stem cells are not frozen. There are two types of allogeneic transplantation. Ablative allogeneic transplantation involves a high dose of chemotherapy. Reduced intensity allogeneic transplantation involves a mild dosage of chemotherapy. The type of allogeneic transplantation used on a patient is dependent on the patient's health and age. In case the patient cannot find a donor, doctors use an umbilical cord blood transplant where they use the cord blood. Another option is parent-child transplant and haplotype mismatch transplant. These more commonly used transplants involve an antigen match ratio of about 50% instead of near 100%. The donor may be a parent, child, or sibling. Both ways or both transplantations are effective in treating leukemia cancer.
Here are some of the applications of stem cells and stem cell therapy. Firstly, they are used to study the specialization of cells, specifically how an organism develops from a fertilized egg. Additionally, they are used to safely study how cells are affected by disease and drugs. In the field of medicine, stem cell therapy solves the problem of minimal organ donors. Stem cells also minimize the risk of an organ being rejected by the body. They can also be used to replace defective or degenerating organs. Here's a list of what stem cells are involved in the treatment of. Firstly, there's spinal cord injuries, stroke, burns, diabetes, cardiovascular diseases, osteoarthritis, rheumatoid arthritis, retinal disease, Huntington's disease, Parkinson's disease, hearing loss, heart disease, and the treatment of some cancers. Stem cells have been applied in the treatment for serious diseases for over 55 years now, especially for cancers. This is how stem cells are applied in cancer treatment. The patient's own adult stem cells can be extracted from bone marrow or peripheral blood before the process of high-dose chemotherapy. They are then transplanted after the treatment in hopes of minimizing the side effects of aggressively high doses of radiation that is involved in the chemotherapy and to help with the regeneration of destroyed blood-forming cells. Stem cells can also be applied in the treatment of some cardiovascular diseases which involve a damaged heart. Firstly, adult stem cells in the patient's bone marrow are removed from the patient's hip. Then the stem cells are injected into the damaged area of the patient's heart. Finally, proteins would be produced that would signal the generation of new heart muscles and blood vessels. Additionally, stem cells can treat type 1 diabetes. Recall that when someone has type 1 diabetes, their immune system targets and destroys the beta cells located in their pancreas. Beta cells are important because they secrete insulin, which is needed for glucose to enter the cells, thus creating energy, in the process known as cellular respiration. Research shows the potential for stem cells to replace the destroyed beta cells into functioning ones that produce insulin, and in turn treating type 1 diabetes.
Stem cell research presents an ethical problem which is affecting the support it receives from government. While stem cells can treat many diseases and injuries, certain religious groups and pro-life supporters object to the destruction of a human embryo. In addition, the government spends billions of dollars in biomedical research even when many people find it unethical. Also, stem cell therapy is still mostly experimental, yet the FDA allows unlicensed clinics to perform therapy. While it's not illegal, many of these treatments are unapproved and have no guarantee of safety. The issue of the body rejecting donated stem cells can have serious consequences, such as organ damage, infection, or the formation of tumors. The use of embryonic and cord stem cells in therapy is still mostly theoretical. For over 50 years, bone marrow transplants have been used to treat leukemia and cancer, and is still the only widely practiced form of stem cell therapy. To prevent rejection by the body, suppression of the immune system may be required. However, this holds the risk of immunodeficiency and disease. Stem cell therapy is still a relatively new technology and has not yet been rigorously tested on humans. Most research is done in labs and on mice. In addition, induced pluripotent cells may not yet be fit for stem cell treatments. Research has shown that these cells have higher rates of cell death and production efficiency than embryonic stem cells. Inducing a somatic cell into pluripotency may also attract retroviruses. Retroviruses are RNA parasites that can change a host nucleotide sequence, which affects gene transcription and consequently form tumors.
In the brief history of stem cell research, 3 presidents have had an influence. In 1993, Bill Clinton allowed National Institutes of Health to fund human embryo research for the first time. Not quite a decade later, in 2001, President Bush restricted federal funding on stem cell research. Obviously a setback. In fact, the 2005 and the 2007 Stem Cell Research Enhancement Acts were both vetoed by Bush. His efforts, however, were in vain because in 2009, Obama lifted his restrictions. Now, with the federal funding restrictions lifted on stem cell research, some cool milestones have been reached. First milestone is related to a disease being cured, and this disease is known as severe combined immunodeficiency, also known as a lymphocytosis, or simply bubble baby disease. Now, this is a genetic disease, so the cure was gene therapy, and this was developed by UCLA researchers. This therapy actually cured 100% of the test of the test clients, which consisted of 9 babies. Nevertheless, all of them were cured, which is really exciting. Now, the other milestone that I'd like to briefly discuss is called mechanoresponsive cell system. And this is a treatment that focuses on destroying metastasized breast cancer cells. And it is looked at in mice. And there's two exciting things about this, which they're kind of linked together. The first is that tissue damage, including lung damage, virtually non-existent because the treatment only attacks cells in the presence of cancerous cells. So again, these are two milestones happening in the year 2017, and it's just a glimpse into the future, into the exciting field of stem cell research.